Good morning, everyone. In today's video lecture, we learn about environmental pollution control under the course of pharmaceutical engineering. So let's start. So first of all, what is environment? Environment can be defined as the natural surroundings of that organism which directly or indirectly influences the growth and development of the organism. It can also be defined as the surroundings in which an organization operates, including air, water, land, natural resource, flora, fauna, humans, and their interrelations. This is the ISO definition. So, what is pollution? Pollution may be defined as contamination of air, water, soil, etc., with undesirable amount of material or heat. When the environment becomes unfit for supporting life, such deterioration is known as environmental pollution. Most pollutants are solid, liquid, gaseous byproduct or waste produced when resource is extracted, processed, or made into products or used. So there can be a lot of waste. What is healthcare waste? So all the waste generated by healthcare establishment, research facilities, and laboratories are healthcare waste. In addition, the waste originating from a minor or scattered source, such as that produced in the course of healthcare undertaken in home, dialysis, insulin injection, etc., 75 to 90% of the waste produced non-risk or general healthcare waste. Remaining 10 to 25% are hazardous and many create variety of health risks. The categories of healthcare waste: infectious waste, pathological waste, sharps, pharmaceutical waste, genotoxic waste, chemical waste, waste with high content of heavy metals, then pressurized containers, radioactive waste. So these are the different categories of healthcare waste. We'll discuss this one by one. I will discuss their source, their hazardous effect, and the prevention mechanism for this waste. First of all, infectious waste. Infectious waste is suspected to contain pathogens like bacteria, viruses, or fungi in sufficient concentration or quantity to cause disease in susceptible host. The category includes cultures and stocks of infectious agents from laboratory work, waste from surgery and autopsies, waste from infectious patient in isolation wards. So these are some infectious waste which obviously contain different bacteria, virus, or parasites or their metabolites. Okay. Then waste that has been in contact with infected patients. These are also infectious waste. Then pathological waste. You need to differentiate among each other. Okay, what is infectious waste? What is chemical waste? What are pathological waste? So pathology. Patho means related to disease. So consists of tissue, organ, body part, human fetus, animal kerasis, bloods, body fluids, etc. Within this category, recognizable human and animal body part are also an waste. The sharps. Items that could cause cut or puncture wound, including needles, hypodermic needle, scalpel and other blades, knives, infusion sets, saws, broken glass, and nails. So these are the sharps. And pharmaceutical waste. Actually, it is different from those of infectious and other pathological waste because pharmaceutical, as the name suggests, it would be regarded to pharmaceutical product like tablet, capsules, uh, liquid dosage form, solid dosage form, etc. It includes expired, unused, slate, or contaminated pharmaceutical products, drugs, vaccines, etc. that are no longer required and need to be disposed of appropriately. Also includes discarded items used in the handling of pharmaceuticals such as bottles, boxes with residues, globes, masks, connecting tubes, and drug vials. All of these are required for manufacturing of pharmaceuticals. So these are the pharmaceutical waste. Genotoxic, as the name suggests, toxic for genetics or genome or gene. So these are highly hazardous, may have mutagenic, teratogenic, carcinogenic properties. Actually, these all of them are related to some genetical disorder. It raises serious safety problems both inside hospital and after disposal and should be given special attention. Genotoxic waste may include certain cytotoxic drug, chemical or radioactive materials, vomit, urine, and feces from patients treated with cytostatic drug. Okay. So source. The source of genotoxic waste are syringe, needles, gauze, vials, packaging, okay, outdated drugs, excess solutions, urine, feces, vomit from patients. So these are some genotoxic waste. Then chemical waste. It consists of discarded solid, liquid, or gaseous chemicals, for example, from diagnostic or experimental work and from cleaning, housekeeping, and disinfecting procedures. It may be hazardous and non-hazardous. In the context of protecting health, it is considered to be hazardous if it has at least one of the following properties, like toxic, corrosive, flammable, reactive, and genotoxic. So if these properties are present, then these are hazardous chemical waste. So some examples are formaldehyde, photographic chemicals. Just go through their uses. Then solvents, inorganic chemicals, okay, organic chemicals like insecticide, rodenticides, etc. Okay, perchloroethylene. Okay, so these are some organic chemicals. So number 7, waste with high content of heavy metals. So heavy metals are very toxic for our cells or for our nucleus or DNA or protein. It represents a subcategory of hazardous chemical waste and are usually highly toxic. Mercury waste are typically generated by spillage from broken clinical equipment, but their volume is decreasing with the substitution of solid-state electronic sensing instrument. Cadmium waste comes mainly from discarded batteries. Okay. Then pressurized container. You know, some aerosol container, meter dose inhaler. Okay, some spray. So this contains some pressurized containers. Okay, for gaseous dosage form. So many types of gas are used in healthcare and are often stored in pressurized cylinder, cartridges, and aerosol cans. Many of these, once empty or of no further use, are reusable, but certain types, notably aerosol can must be disposed of. So most common gases used in healthcare are anesthetic gases, ethylene oxide, oxygen, compressed air, then radioactive waste. So radiations of interest in medicine include X-ray, alpha and beta particles, gamma ray emitted by radioactive substances. An important practical differences between this type of radiation is that X-ray from X-ray tubes are emitted only when generating equipment is switched on, whereas radiation from the radionuclide can never be switched off and can be avoided only by shielding the materials. So there are some major and minor sources of healthcare waste. So major sources are hospital, other healthcare establishment, related laboratories and research centers, mortuary, animal research and testing, blood bank, nursing homes. And minor sources are some healthcare establishment like physicians' office, dental clinics, acupuncturists, specialized healthcare establishment and institutions. Okay, non-health activities involving intravenous and subcutaneous interventions, then ambulance service, home treatment. So these are some minor sources of healthcare waste. I've already discussed this. Just go through this. Some medical wards, operating theaters, okay, other healthcare units. These are the sources. Then laboratories, pharmaceutical and chemical stores, support units. So what are the hazards of healthcare waste? We have already learned about some sources and categories of healthcare wastes. Now we'll discuss the hazards. So exposure to hazardous healthcare waste can result in disease or injury. The hazardous nature of healthcare waste may be due to one or more of the following characteristics. First of all, it contains infectious agent, it is genotoxic, it contains toxic or hazardous chemical or pharmaceuticals, it is radioactive, it contains sharps. Persons at risk are healthcare waste, medical doctors, patient in healthcare establishment, visitors, workers. Okay, these are persons are at risk. So there are some hazards from infectious waste and sharps. So puncture, inhalation, ingestion, okay, through mucous membrane. So different types of infection and their causative organism and transmission vehicles has been given here. You can go through this. So hazards from chemical and pharmaceutical waste. Intoxication can result from absorption of the chemical or pharmaceutical through skin and mucous membrane or inhalation or ingestion. Okay. Burns, injuries to skin, eye, mucous membrane of the airways. Hazards from genotoxic waste. Actually, these genotoxic substances may cause um, some genetical changes, okay, and genetical disorders within our body. So hazards from radioactive waste. So it ranges from headache, dizziness, vomiting to much more serious problem. Because radioactive waste, like certain pharmaceutical waste, is genotoxic, it may also affect genetic material. So handling of a highly active source like certain sealed sources from diagnostic instrument may cause much more severe injuries, as destruction of tissues. Okay, so these are some hazardous material. Now, treatment and disposal technologies for healthcare waste. Actually, in pharmaceutical engineering, we learn different treatment and disposal technologies. So, what are the common technologies for management of this healthcare waste? We learn this one by one. So these are the methods like incineration, chemical disinfection, wet and dry thermal treatment, microwave irradiation, land disposal, and inertization. So we'll discuss the methods and their advantages and disadvantages one by one. Let's start. First of all, incineration principle. High temperature dry oxidation process that reduces organic and combustible waste to organic incombustible matter with significant reduction of waste volume and weight. So combustion efficiency is 99.9%. Temperature is primary chamber 800 degrees Celsius. Secondary chamber is 1050 degrees Celsius. Capacity is 200 kg per day or 1 ton per day. No pre-treatment, waste converted as gas and heat. So you can see here waste product has been kept here. Okay, then it has been sent here, and incineration has been takes place here. So after incineration, the gas is used for what? Is used for electricity generation. Okay, the turbine is rotated and electricity is generated. And you'll find some ash from here, and you'll collect the ash. And the gas also be collected and will be wiped out by this way. And wastewater and ash for treatment or disposal go through here. So you can see how we utilized our waste for generation of electricity. And the remaining waste after the treatment has been sent for disposal. These are common process for incineration. Actual incineration is taking place here. Okay, this is the actual location, and other the screw pads and bag house are just for emission of the gases. So what to burn? Infectious waste, pathological waste, pharmaceutical and chemical waste. Okay. And required waste characteristics are heating value is above 350 kilocalorie per kg, then content of combustible matter above 60%, and moisture content below 30%. And what should not be burnt? Pressurized gas container because it may explode. Then radioactive waste because they may disperse the radiation. Then halogenated plastic like PVC, it can exhaust gases containing harmful hydrochloric acid and dioxin. Then waste with high mercury or cadmium content like broken thermometer, used batteries, etc. Then sealed ampoules and ampoules containing heavy metals. Then chemical disinfection. Chemicals used to kill or inactive pathogen for treatment of liquid infection waste such as blood, urine, stool, or hospital sewage. Microbial culture, solid waste pre-treated chemically only after shredding. Conversion to small pieces increases surface contact with disinfectant. Chemical used includes 10% sodium hypochlorite, 5% phenol, formaldehyde, glutaraldehyde, ethylene oxide. Then neutralize chemical before draining out. Then wet and dried thermal treatment. It's like some micro oven. Okay, just thermal treatment is applied here. For wet thermal treatment, steam is applied. Okay, common laboratory practice for disposal. And for dry thermal treatment, state the waste heat in treating device, then 80 to 90% reduction in waste volume. So large scale autoclave offsite treatment principle is 121 degrees Celsius temperature under pressure 15 lbps for 20 minutes. So this is for autoclaving. Then dry thermal treatment, screw-free technology. Waste is straight to particle of 2.5 centimeter in size, then added to a rotating auger, then shredded waste left in auger for 20 minutes. Residues are compacted. The microwave principle is just microwave. Microwaves are used at a particular frequency and wavelength. Shredded waste chamber with series of microwave generator, 20 minutes at least, and capacity up to 250 kilogram per hour. Then land disposal. This is one of the most commonly used method for this. We need to learn this very carefully. Sanitary landfills only, especially designed and constructed, no treat waste, prevent underground water contamination, then facilitate treatment of on-site leachates. So land disposal, healthcare waste should not be deposited on or around open dumps. The risk of either people or animal coming into the contact with infectious pathogens is obvious, with the further risk of subsequent disease transmission, either directly through wounds and inhalation or ingestion, or directly through the food chain or pathological host species. So you can see here how land disposal is carried out. So it can be of two types, like deep burial pit and sharp pits. Deep burial pits, trenches of at least 2 meter deep, each time which is added and 10 centimeter layer of the soil to be overlaid. Let's cover the pit, and it should be away from habitat. And sharp pit, all sides to cemented and concreted. It's not as like as deep burial pit, which is open. Sharp pit is not open as open as that of deep burial pit. Only a pipe is introduced. All sides are cemented or concreted. A cylindrical pipe. Pipe is introduced of 4 centimeter diameter, fixed at ceiling through which shafts are deposited. We'll see this in the figure. This is the deep burial pit. You can see how openly this has been kept. So waste product has been kept here, and this is open. Okay, but in case of sharp pits, you can see here this is cemented or sealed, and here is a steel pipe 4 centimeter through which you will apply your waste product. You can see your waste product has been kept by this way, and that is why its location is in here. So this is the basic difference between the sharp pits and the burial pit. And next one is inertization. Suitable for pharmacological and incineration ash, a mixture of water, lime, and cement added to the waste. For example, pharmaceutical waste is mixed up with lime, 50% cement, and 5% water. A homogeneous mass formed cube or pellets are made, transported to the suitable site. Alternatively, the homogeneous mixture transported in liquid state to landfill. So it shows the different disposal methods and their advantages and disadvantages that I have described earlier. So for incineration, it is a very high efficiency and incomplete destruction of cytotoxic, and it's high cost. The chemical disinfection, okay, it's very inexpensive, okay, and highly efficient because of chemical reaction takes place here, but it's not suitable for pharmaceutical or chemical waste, okay, and chemicals are also hazardous. Then autoclave, within a very big autoclave machine, you applied your waste. That means it's low investment and operating cost, very handy. Okay. But main important thing is that it needs trained personnel and to not educate for waste that are stream impermeable. The microwave, drastic reduction in waste volume and weight. It is environmentally safe. Then potential operation and maintenance problem. Then land disposal, that is very low cost and safe, and used only after pretreatment of waste. So this is the overall pros and cons of our different methods, and this is the overall lecture from this uh, lecture. If you have any confusion, please inform me in the comment section. Thank you.